Yeah, thanks very much for the very kind introduction, and thanks for the invitation to come to Frankfurt. I think the last time I was here was in 1987, when I just passed my driver license on the 4th attempt, and in a celebration of this achievement, I did my first solo trip in an automobile, and it led me here to Frankfurt. So today I want to talk to you about this topic it's just mentioned, and my hope is that even if you don't care about soil or terrestrial ecosystems or global change, you will still find something interesting in that talk, even though I hope that at least some of you care about this and the combination of that. Because I'm talking about something much more fundamental, I think, that would be broadly applicable to maybe many of the things that you do here, and as I've already heard throughout the day. But I'm going to start out with a little bit of this setting, why we care about soils. So our soils, as it turns out, are a really major target of all drivers of global change, in many cases simply because of gravity. You know, things, whatever we put in the environment, eventually they end up in the soil. Of course, eventually also in the oceans, but primarily in terrestrial ecosystems, soils are a major target. And if we don't take care of our soils, then we basically start feedback cycles that tend to make things worse. So soils can be part of the solution, but they can also be part of making things worse if we don't take care of soil health. So first of all, I want to talk to you about the scientific question I have picked for today, which is the one that currently excites me the most, and where also quite a few people in the lab work on. And that is the topic of global change. Now, when you talk about global change and terrestrial ecosystems, and you ask people what are the important factors of global change, you get this answer. The famous big four are warming, atmospheric nitrogen deposition, drought, and elevated atmospheric carbon dioxide. The answer will be different if you work in aquatic and marine systems, but for terrestrial ecosystems, these are the big four. And we know this because we also verified that this is really what most research is being done on. Now, a couple years ago, I read this paper by Emily Bernhardt and others. And in this paper, they argued that ecologists should really also care about synthetic organic chemicals and chemical pollution. We talked about this earlier today, where most ecologists wouldn't touch this topic because as ecologists we basically take it up with the milk of the discipline that we want to work in beautiful untouched places, not in places where things don't work anymore. So most ecologists don't work with synthetic organic chemicals because that's what ecotoxicologists do. But I think we definitely need to add this huge range of synthetic organic chemicals to the list of global change factors. Now, we also in the lab worked on microplastic actually for a number of years. We started in 2012, was the first paper on that topic, and I've since worked on this intensively for quite a while. And actually one of the first papers we wrote was also that microplastic should be considered a factor of global change for reasons that it occurs basically globally, is clearly linked to human activity, and it affects biota. So basically now we add microplastic to this list. The list is growing. And when you think about it just a little bit more, you can come up with quite a few more factors. There's salinity, there's soil compaction, there's various heavy metals, there's artificial light at night, which we now also know affects soil. There's invasive species, there's noise pollution. And basically, when you think about global change in terrestrial ecosystems, and especially the soils, your answer should be there is a lot of factors, not just these four factors. And it is also very likely that these factors all co-occur. And they, in their combination, pose a threat to soil health. You could also say this is a high dimensionality problem because we have very many factors acting at the same time. So we think this is the reality. And I think there's plenty of evidence that this is the case, that there's in any given system always quite a range of factors that act on the system. So what do we know about this? What do we know about the simultaneous action of many global change factors? So we did a systematic mapping exercise a number of years ago, and we looked at a few thousand papers, the literature basically on that topic of global change done on soils with an experimental approach, not observational, but with experiments. And we simply counted what is the number of factors that are being examined in any one study. And laser pointer? Yeah. And basically, unsurprisingly, when we counted the number of factors that are being examined in any one paper, you would have guessed that also, most of the papers deal with one factor of global change. And then there's already quite a drop to when papers dealing with two factors. And I could barely see the three factors, and basically the four factors is almost non-existent. So basically, this is the reality, what we know about the situation. We know there's very many factors acting concurrently, simultaneously affecting systems at the same time. Yet, when we look at how this is being experimentally addressed, we don't see that. We see people work for very good reason, that I'll talk about next, with basically one or two factors. We also don't see that this pattern has changed at all through time. Same colors as over there. So like one factor becoming maybe even slightly more dominant over time, and the other factor numbers basically don't make an appearance. So this trend has also not changed over time. So to drive this point home with one big number is 98.2% of papers ever been published that describe the effects of global change on soil and terrestrial ecosystems using experiments have only included one or two factors, which is another way of saying we don't know what really happens, at least from experiments, which are our best way to know about the world and to establish causality, what really happens when we have more than two factors. So you could ask yourself, why is this experimental work not being done? So there's a couple of reasons that I came up with. Maybe you can come up with others. One, there is logistical constraints, like it is very difficult for labs to gear up, to have all these exposure systems and to have the background to do this kind of work. There's also a psychological effect that very many people rely on saying, well, we captured the gist of it by just looking at the dominant four factors, the big four factors, and the rest is just basically background noise. Many people may think that, for which there's no evidence whatsoever, but I think that this is a psychological thing, that people think that they have captured the essence by studying just the big four. There's a fair amount of research compartmentalization in global change biology. Global change biology is a huge field. And so people working on invasive plants, for example, go to often different meetings that people that work on elevated atmospheric CO2, for example. And they go to different meetings again that people work on atmospheric nitrogen deposition. So there is quite a siloing in that kind of research, so that labs basically focus on certain factors. There are people that work, labs that focus on elevated CO2 effects, and there are labs that focus, let's say, on invasive plants. But there is also one very, very practical reason for why these experiments are not being done, and this is known as the combinatorial explosion problem. That means if you say, let's say you have 10 factors, and you wanted to examine every combination of all these factors with each other, the common tool you would use for that is a factorial experiment, right? So if you have a 2 by 2 factorial experiment, you have 4 treatment combinations. You have each factor just on and off or low and high. This is fine. If you add another factor, this multiplies by 2, right? Then you have 8 treatment combinations. Then you have 16, 32, 64, 128, 256, 512. And if you have 10 factors, you have 1,024 treatment combinations. And then, of course, you need to replicate that stuff. Huh? So, and that's impossible. So basically, this is not possible to do. You cannot use a complete factorial design. This kind of tool that we use to look at factor interactions completely fails. In fact, if you look at a statistics or experimental design textbook, they say, do not do these experiments with many factors, because also in the end, you cannot interpret these interaction terms. And your power to detect interactions goes down the crapper. So basically, this is all known as the combinatorial explosion problem. If you deal with more factors, you have many more treatment combinations that you can reasonably deal with, at least in ecology. And well, this is the contrast that I've just tried to work out for you. So in Experimental studies, we focus on mechanism, of course, for good reason, and therefore we routinely examine only one or few factors at a time that we then study in great detail. But in observational studies in ecology, we very routinely acknowledge that the world is affected by more than one factor. Nobody would do a gradient study and just measure, let's say, precipitation, if this is a precipitation gradient. You would also measure all kinds of soil properties. You would measure the vegetation, nutrient status, pH, other climate variables, and whatever. And you would tease out the factors of interest in your observational study. So basically, in ecology, in observational studies, we very well know the world is complicated, is influenced, anything is influenced at any one time by sometimes dozens of factors. But we wanted to do experiments. So what did we do? So this is an experiment that I did. It was the first time I went into the lab in many, many years, because I had this great idea, and I presented it like all ideas, I presented in a lab meeting, and then I asked, and who wants to do it? Everybody avoided eye contact. So I was like, nobody wanted to do it, because it seemed like a pretty crazy idea, and most likely it wouldn't work. And so I ended up setting up myself, but of course with then a substantial help by other people. This is what the experiment looked like, at least there was a part of it, was set up with these small little microcosms with like 30 grams of living soil in there and exposed to various treatments. What you can see are, for example, the heating treatments. And so the first part of what we did is just look at the factors by themselves in isolation. 10 different factors, as trailered in the previous part of the presentation. So we have control, and basically that dashed line going across, this is the control value. Here you have my favorite response variable, water-stable soil aggregation, so the ability of soil to withstand a disintegrating force and not crumble and fall apart. Basically, it's just a measure of soil stability, if you want. And here we have the 10 factors. We have drought, nitrogen deposition, temperature, microplastic, glyphosate, antibiotic, fungicide, copper, salinity, and insecticide. They were all chosen for various reasons, and they were all added at a concentration that you expect in the environment. And so what you see is, well, a bunch of these factors affected that variable of interest here negatively. It's about 3 or so. And a bunch of them affected it positively. It's about 4. And a bunch of them had no effect, also about 3. That will become important later, that there was about as many positive as negative as neutral responses. So they were not all negative. This is actually quite normal from global change. We know many factors have nominally positive effects on systems. Okay, this part is simple, but then what did we do for the interactions? Knowing full well we couldn't do a fully factorial experiment because it would require an unreasonable amount of experimental units. So this is what we did. We asked a different question. Well, I asked a different question. And this is the solution, or one possible solution to this high dimensionality issue. So I imagined I had a pool of 10 factors in there. And when I, for example, want to figure out what happens when I have 2 factors in the experiment, I sample randomly for every replicate of the factor level 2 by drawing from that pool of 10 factors. Okay? So this is a replicate, this is a replicate, this is one, and there were 10 replicates. So maybe the first replicate of factor level 2 is microplastic and warming. Maybe the next one is insecticide and drought. Maybe the next one is whatever, antibiotic and antifungal agent. So they're all different. And now you know why nobody wanted to do this experiment, right? Because it seems like almost stupid. Huh? So I mean, you look at 2 factors, what do 2 factors do? But every replicate of factor level 2 is different. Most likely, because it's done by random draws from that pool of 10 factors. So, but that's important to understand. So basically, every factor level was determined by the number of factors, and then every replicate most likely differed in the actual composition and identity of the factors, such that what they had all in common was that there was 2, but it was completely random which 2. And by doing that, this is an experimental design that some of you may be familiar with because it's used in biodiversity ecosystem function experiments, for example, with plant communities, where this is directly stolen from. If you want to know what is the effect of richness of plants, you do exactly that. You try to de-emphasize the importance of the identity and the composition of the plant community, and instead ask a question about richness, which is the number of things. And this is just exactly that same idea, but applied to global change factors rather than plant species. Okay, so this is what we did, and the expectation was actually, this is going to be all over the place, because you're comparing literally apples and oranges, even within one factor level. As I said, this is like totally random what's in each one of those experimental units. However, this is just a reminder, what you've just seen in the previous slide, this was not the case. So you have already seen this part here. This is just the single factor responses to each of those factors applied in isolation. And here is this random draws from a pool story on the right. So factor level 1 is just all of these data together, because this is also one factor. So you can add them all basically in here. This is one factor active. And then we have 2, 5, 8, and all 10. There was only one way to sample 10 out of the 10. We've subsequently done this again, and then we had a pool of 12. So they're not all identical, but it doesn't make a difference. It's all the same result. Basically, this was not random at all. I mean, this is really the shocker, that it basically is a pretty straight line here down. So the more factors you add, the worse this is for soil aggregation. Now was the part when you needed to remember that the single factor effects were actually equally, by chance it wasn't planned, were equally frequently positive or negative or neutral. So it is very much not because you're just adding increasing amounts of negative stuff. That does not explain that. Actually, what explains this, we still don't know. Yeah? But this is pretty phenomenological, and since we've repeated this many, many times, and it's always got the same result. But basically, this is what comes out. We don't really exactly know why. We're working on this now. But basically, it was not just adding more negative effects up, because there were as many positive as negative as neutral effects with the single factors that we're just randomly combining here, and we get this increasing decline. Yeah, people smarter than me did various statistics, so you can see that when we do null hypothesis testing, the experimental results, which are these black dots here, this is exactly the same as over here, deviate increasingly and then eventually also very, very much statistically significantly from various null model expectations, additive, multiplicative, and dominative, which means for every one of the replicates, we, of course, even though this was done randomly, we know which factors were picked, and we could formulate an expectation for what the result should be in the absence of any interactions. There's 3 ways you can do this, mostly. These 3 null models, you just add the effect, you add the proportional effect, or you say the dominant factor wins. These are these 3 null models that basically say there's no interaction. So basically, the fact that the null models don't fit the data at all means there is interactions. And also, we tested for the number of factors as a continuous variable, basically as a factor, as a categorical variable. This was highly significant. And just knowing the number of factors, we could explain over 50% of the variability in the data set, which when you think about what this really is pretty astonishing, because this was just all kinds of random combinations of factors in the end. And of course, when we added more information, like when we added knowledge about factor identity, the variability explained jumps way up. And now we also measured other things. And one of the things that was really surprising, one of the things we measured next was water repellency, which is basically, you all notice from your houseplants, when you add some water to your houseplant, you haven't watered in a while, the water pearls off, so the soil becomes hydrophobic. And this was super interesting because there was no effect of any of the single factors except for the very well-known effect of drought that caused a little bit of an increase in water repellency. This is well known for the sandy soils around Berlin. But look what happens here with the factor combinations. Basically, from a factor combination of about 5 on, there was this big increase in soil hydrophobicity, something that you wouldn't have predicted from anything in the single factors because there is no information contained in the single factors that would allow you to predict this increase. And you know, I can remind you that 5 factors is exactly that nobody looks at. And it happened to be that from 5 factors on, we saw this big increase in water repellency. Of course, null moles don't work at all because there's not much information contained in the single factor effect sizes and directions to predict that. And then, of course, the amount of variability explained is very low. In other words, we couldn't see this coming. This is what you would call an ecological surprise. It's like when you combine very many factors, then there is a completely unexpected result. This could be due to a number of things. In the end, we don't know what causes it, but there's a bunch of hypotheses. You have seen the top 2 panels. I've just shown them to you. We measured other things like decomposition rate and respiration rate, and they all show more or less the same pattern of this increasing decline with an increasing number of factors. We work with fungi in my lab, so we looked for fungal diversity by high-throughput sequencing and ASV richness, which you can think of something like, but not really a species richness. You can also see that there is a quite sharp decline in the number of fungal species that drop off. So species went extinct in these small microcosms. For those of you who care about fungi, the losses were very much non-random. What we lost were the Basidiomycota predominantly, rather than Ascomycota or other phyla. There was directional change, beautiful linear pattern basically in composition and also dispersion, the difference from the centroid. So in all of these variables that we measured, we saw this straight decline along with the number of factors. Now, you might think this maybe was just a one-off result because something was funny in these microcosms and so on and so forth. Something had to do with like the number of factors, with the particular identity of factors that we used in this experiment, or something else about the experiment that we did. We've since repeated this actually several times, multiple times. We're still repeating it many, many times. One of them is published already. This was done with a different soil, actually with an agricultural soil rather than a meadow soil. That was, the meadow soil was from quite a bit outside of Berlin, so it was a pretty pristine soil, more or less. Of course, there's no pristine soil, but you know what I mean. And there was also different factors here. There was, for example, also soil compaction and other variables. Other factors were used here, and we still saw the same response. This experiment was twice as big, actually, because we did it at low and high soil biodiversity, because we asked, can biodiversity buffer for the effects of an increasing number of factors that impinge on that soil? And the answer is yes, up to a point. So in the beginning, there was quite a difference in the effect size comparing the different biodiversity treatments. But if you add more and more of these factors, basically these curves here, the various response variables, they converge. So in the beginning, at the low levels of factors, like 1 or 2, or 0, of course, there was a big effect of soil biodiversity. But these effect differences between low and high soil biodiversity disappeared with an increasing number of factors, of course, because you lose biodiversity from the high biodiversity treatments. Then we were very curious if we could see that same kind of pattern also in the field. So we set up this field experiment. It's my first field experiment. I would not recommend that as the first experiment to set up. But this was set up just a few minutes walk from our institute in Berlin. And here you also have 10 treatments. It's actually the same design that I showed you, slight difference in factors. And what you can see here basically are the climate change treatments. So we have open top chambers for passive warming of about 1.5 degrees. And we have rainout shelters that intercept about 50% of the incoming precipitation. The other 8 factors were added to soil, like tire wear particles, salinity, copper, insecticide, fungicide, antibiotic, things like that. And then we use 2, 4, 6, 8, and all 10. So the same design that you've just seen. Looks pretty from the top. And also this is now running in June. Well, in October it will be 3 years. So we'll have probably a final harvest this June. And the data are currently being written up. But what I can tell you basically is that we see very much that same response for plant diversity. So we have a massive loss of plant species along that gradient of factors from 0 to 1, 2, 4, 8, and 10. And alongside that, we also see differences in other parameters that are unrelated to soil. For example, we see pollinator visits decline. We had about 300 undergraduate students in that field measuring pollinator visits, not all at the same time, but in shifts. And so we see also that. We see flower numbers diminishing also along the factor gradient. We see leaf pathogens increasing on certain plants along the factor gradient, and so on and so forth. So, but we're still harvesting one more time, and then now we can be sure that it's not just short-term responses, and we know that it's not just an artifact of having done this to a small volume of soil, because we also see this at the level of the plant community. I had shown you this slide before here, where I pointed out to you that in ecology we basically have this big two approaches to empirical research. One is experimental, which I've just shown you data for, but there's also observational studies. And I've told you in observational studies we very often measure data for a lot of variables. And so I was just curious if we could tap into some of the observational data that had already been collected and see if in observational data we can also see the signature of the number of factors. But now you're dealing with data that's not from an experiment, and you're dealing with data that's continuous. And so you now need to translate that to the number of factors. So we needed to do 2 tricks. One trick is we needed to convert the continuous data to numbers of factors. And that means what we used is a thresholding approach. We said like, okay, for this continuously measured variable, we count it as on if it exceeds a certain threshold. And we count it as off if it doesn't. We do that for all factors. This is observational data, so it has very many statistical problems, for example, multicollinearity. So we needed to combine factors in a way that there is no multicollinearity problem. This was the second trick. So what I show you the results for is a substantially lower number of factors that you would expect, because we had to group factors together to avoid the multicollinearity problem. And so basically this is what we did. This was a global data set using forget now exactly, I think it was 200 different ecosystems across the globe. And of course, when you have a thresholding approach, you have to always pick which threshold do you use. And the answer to that is you use all thresholds. And this is what you see in the top figure. This is from 3 different global surveys. And basically, the lighter that color is, the higher level threshold you have to pass in order to count a factor as on. And so you can see this is the number of factors, basically, that cross the certain threshold. And here you have something like multifunctionality, including also diversity, all thrown into one variable. It's also been separated out in the paper if you want to look at it. And basically, as you exceed increasingly higher level threshold, it's a little bit abstract, which means you count factors as on if they are much, much higher up in the level of your data set, like in terms of the percentile. You count like maybe only the 75th percentile as on, or as opposed to like the 5th percentile, which would be very, very low. So the higher you go, the higher that threshold is, the more negative that curve becomes. You can see that for all these 3 different independently conducted global surveys. So that means the higher the number of factors, this number of factors is much lower than the number of variables measured because we had to group them together to avoid this multicollinearity problem. You can see there is also this decline in biodiversity and ecosystem process rates that were measured. There were a lot of them measured in these studies. We have a lot of data. And the different surveys differed in the number of variables that were measured. For some, microplastic was measured. For some, it was pesticides that were measured. But you basically put that together, you always see the same pattern. And basically also the models that included that high-level threshold exceedance, they were always selected by the model selection procedure. So this was the signature of the number of factors, if you will, was also visible in this global observational data set. So it seems to, the number of factors acting on a system seems to explain a unique proportion of the variability in this global data set. This is the signature of the number of factors. And if you will, this is the signature of unknown higher level interactions among all these variables. So we have evidence not just from variety of experimental approaches, but also from observational work. Okay, so the final part is, what does this all mean? That means, first of all, we have a lot to do because we know, which you know from the systematic mapping, one of the first slides that I showed you, we know very, very little about what happens when you have a very large number of factors acting simultaneously. And I think we need to think much more about this high dimensionality because there could be all these surprises lurking that nobody ever sees because they didn't look at very high levels of factors in their experiments. And we need many new ideas and many new approaches. And in fact, we're currently in the lab, we have quite a number of people that look at different ways of doing that, playing with this idea of the number of factors, the identity, the composition, the diversity of factors. So we have a lot of experiments finishing and running right now that play further with this idea. Also something that I was very interested in trying to bring some order into these global change factors is to try to classify them. And I'll show you some empirical results from this just after that. So this is basically a project where I looked at 30 different factors of global change. They are arrayed around this circle here. So there are physical factors, chemical factors, combined physical, chemical factors, and biological factors. 30 factors of global change that I each asked the same question, 30 questions. So it's a matrix of basically 900 cells. I asked each global change factor the following question: are you physical? If you're physical, what are you? Are you energy, or are you some other force? If you are chemical, are you abiotic? Are you inorganic? Are you organic? And so on and so forth. So I each asked all of these a number of questions. Here is sort of the questions zooming in here. What is your nature? What is your proximate effect direction? What is your effect mechanism? And what are some key properties? Basically, it's a trait-based approach to global change factors. Basically collected mental data on 30 traits for each of the global change factors. What's new is that they're now all on the same plane. All of these factors that are normally dealt with completely separately, also normally by different people, they are now all in the same table. And incidentally, the little shading you see here, these are the data. They are just zeros and ones. These are the answers to all the questions of the 900 cells here. Now, when you have that, what you can do is do a classification. And this is what comes out. This is a classification of the 30 global change factors based on these traits. Now, 10 of these 30 global change factors were the ones, not by coincidence, that we used in this experiment I just told you about in the very beginning. So if we regroup that correlogram, it's this here. This is basically the similarity or dissimilarity tree. Now, and this here on the far right are all the experimental data I showed you from this experiment on a specific instantiation of these particular factors, a certain strength. It was just not heavy metal, but it was a specific heavy metal, namely copper, at a particular concentration, in a particular soil, in a particular experiment. And so then we just basically for shits and giggles, we looked if these two trees have a higher correspondence than you would be expecting by chance alone, and they do. They are, you can see it sort of from the color coding a little bit. Of course, it's not perfect. This would be terrible. We would stop to do experiments. But this is basically just armchair thinking about these factors on a very fundamental level. And these are the experimental results. And there's some correspondence between the two. And so we have now used this in terms of factor similarity for the next generation of experiments, where we basically do communities of global change factors with increasing and decreasing similarity, and we see very strong effects of that on the outcomes. So, to wrap this up, what can we do basically to help? First of all, like to always have something positive, because in the end, this is all pretty depressing. Basically, this talk says, you think it's bad? It's even worse. So it's very nice to say also something positive, because I'm a pretty positive person. So basically what this means is, first of all, it's always good to know when an effect occurs. It's very good to know that just the sheer number of factors impinging and acting on a system has a negative effect. What that means as a possible silver lining, even though I don't know, but we're currently working on this, is that if you reduce just the number of factors, you might already help. And basically, what our work says is it doesn't even matter which factor you reduce, it's the number of factors that drives the effect. So you could basically reduce any factor, and maybe you would be going back along this curve. We don't really know that exactly, because there could be hysteresis effects, so the journey there could be different from the journey back. But it at least is an idea to work on. The next thing is we really need to unravel the mechanisms on these interactions. And so I've done two things. First of all was the realization that some of these factors act on completely different levels. We have some factors that maybe act on the level of the landscape, and we have other factors that enter the system basically at the level of, like, say, the individual plant. And they all have effect propagations from the top down and from the bottom up. And since they don't all work on the same scale, it's actually relatively new insight, they can also produce interactions by meeting somewhere along the way. And also it turns out a lot of these interactions among different factors don't have names. We don't have names for factor interactions, but there's some factor interactions that you can give names. I tried that in this one paper. Like, for example, if you have a drought, of course you have the absence of water, but in soil, if you have less water, you increase the effective concentration of any chemical. So you can think of drought as a concentration amplifier. And so you can use this name. And so I've proposed a bunch of other names that describe and give names to these interactions of factors, at least at a pairwise level. And finally, I think what is very important, and I'm getting more and more passionate about this point here, is that how we communicate about this is, I think, very important. And I think it is very important to unify the message here. Because people have, like, a limited pool of worry. They can only worry about so many things. If you say, like, oh, you have to worry about this, and you have to worry about microplastic, and you have to worry about 350,000 registered chemicals that are in the environment, people will close down. When, in fact, a part of the reason for this entire exercise in this research program has been to basically say this is all global change biology. We have a word for that. It's called global change, or global environmental change. And what I mean by that is human-caused global environmental change. This is a technical term. Global change, if you go to anybody on the street here, or in the US or anywhere, they will not know this word. This is just known in our bubble. That was kind of news to me. I found it out last year. Climate change, everybody knows. You can go to the supermarket and ask anybody, have you heard of climate change or Klimawandel? And they will say, yeah, sure. But they have not heard of global change. But we have a word, we can use that to communicate to people that this is all part of the human impact on this planet. It's actually one thing. It is not 1,000 different things. It's one thing. So it is, I think, very important how we communicate about that. And that's kind of the mission that I'm currently busy with. And this is all I want to talk to you about today. So I'd like to thank these fine funding agencies for their support. Much of that work was funded by an ERC Advanced grant, and the wonderful people in my lab, and you for your attention. Thanks.